Good morning, everyone. I don't know if you can hear me. Okay. Thank you all for coming this Saturday morning. It's a pleasure to have you here. And it's my pleasure to introduce Dr. June Nasrallah, FAS guest today. I met June a few years ago, and then yesterday was the second time we meet. And I know she's a brilliant scientist, accomplished, well-known, and when I told her I was going to be presenting her, she said that's going to be easy. It was AUB first, then Cornell all the way. And to summarize 46 years of research work and efforts in academia. So I'd like to take a bit more time, but I've been warned not to do that beyond the 3 minutes. So I'll try to limit myself to that. June Nasrallah was born and raised as June Bowman in Beirut. She went to school at the Collège Protestant for most of her school years, and then spent 1 year at IC before joining AUB. Her love for science, and specifically plant biology, was nurtured during her school years. June Nasrallah continued studying biology at AUB and received her BS degree in 1970. It was during these years that she met her husband, Dr. Mikhail Nasrallah, and together they started a long journey that's still going strong in academia and research. Dr. Nasrallah joined the lab of an eminent geneticist at Cornell, Dr. Adrian Serb, and received her PhD in 1977. During these years, she returned in 1975 to AUB, and specifically to the biology department as a TA. This was in 1975, but during that year, you all know that the Civil War broke out, and she returned to Cornell and completed her PhD. Talking to her yesterday, she told me that by that time she had 2 kids to raise and research to attend to, which was not an easy path to tread. She recalls that her mentor, Adrian Serb, at the time told her that she had to stay in research and insisted that she comes into the lab and work on whatever she pleases and at her own time. She said it was this and her interest in science that kept her going. Mind you, during this time, she managed to publish 5 papers. Her research focus is in plant reproduction, self-incompatibility, and receptor-ligand interaction. I won't dwell much on the biology. I will leave this to her. But Dean McGreevy, yesterday in his address, he talked about Darwin and the Lewis affair at AUB. Well, Dr. Nasrallah carried the torch further in support of Darwin, I guess. In a biography on June Nasrallah, Kristen Brownlee writes, Darwin observed more than 100 years ago the so-called self-incompatible plants. To put it simply, that is plants that do not self-pollinate and can only become fertilized and produce seeds with pollen from related plants. The genetic mechanisms underlying this phenomena remained an enigma until the 1980s, when June Nasrallah and her colleagues demonstrated that recognition or self-related pollen is determined by specific interactions between a receptor in the stigma, which is the female reproductive organ in a plant that captures the pollen, and its ligand, which is expressed solely in pollen. These studies not only provided a basic foundation for plant genetics and evolutionary research, but also have the capacity to transform and improve plant breeding techniques. In recognition of her career, long career, and research leading up to this accomplishment, Nasrallah was elected to the National Academy of Sciences in 2003 in the US, which is a highly prestigious recognition. She also received the Martin Gibbs Medal in 2003 from the American Society of Plant Biology. She was named the Barbara McIntosh Professor of Plant Biology at Cornell University in 2004 and was a founding member of the Lebanese Academy of Sciences. One cannot talk about June Nasrallah without acknowledging her lifelong partnership with her husband / co-investigator, Dr. Mikhail Nasrallah. Together, the Nasrallah team continued to work on elucidating the complexity of self-incompatibility. June hinted that this unique partnership could be one reason for the Nasrallah's continuing success. She states, "It's extremely advantageous having someone so close to talk about the research." She said, "It's like a 24-hour immersion in the science." This partnership extends beyond their day-to-day life in the USA. Yesterday, in talking to her, I've learned that for the past several years, the Nasrallahs returned to their hometown close to Rashaya, work on their vineyards and orchards, making wine and olive oil and shipping it back to Cornell. At AUB, we'd like to invite you to an additional stop during your annual visit. We'd love to see you here at AUB, your alma mater, every year. Hopefully, we can do this next year and on. Dr. June Nasrallah will be talking to us about biology for a rapidly changing world. Dr. Nasrallah.
Thank you very much for invitation and for the kind introduction. I apologize in advance. I seem to have a little some allergy. So in any case, 150 years ago, the year 1866, the founding of AUB, was a landmark year for biological science. In that year, Gregor Mendel, a monk working in Brno, published his Laws of Inheritance based on his work on peas, and Charles Darwin refined his ideas on the origin of species. They were contemporaries, but not necessarily aware or appreciative of each other's work. But at any rate, the two of them laid the foundations for large principles in science that apply to all living organisms. In his book on the origin of species, which consisted of 502 pages, there was one figure, possibly the most influential figure in biology. It showed a branching tree of life that depicted the origin of new life forms by descent from a common ancestor. While the 1800s were characterized by the elaboration of large basic principles of biology, the 1900s were characterized by advances in DNA science. In 1944, and these are just a couple of highlights, in 1944, Avery demonstrated that it was DNA, not protein, that was the genetic material. In 1953, Watson and Crick reported on their solving of the DNA structure, which consists of a double helix with each strand consisting of a string of 4 letters, AGCT. This work and a lot more work led to the elaboration of the central dogma of biology, shown here, which states that DNA replicates itself as the cells divide. It's transcribed into a messenger RNA, which is translated into protein. And this work and subsequent work into the 1980s completed the transformation of genetics, which was rediscovered in the early 1900s, into molecular genetics and recombinant DNA, which allowed the joining of 2 pieces of DNA from any source. So in my talk, I'm going to talk a little bit about our research on self-incompatibility that was just mentioned. And it's a secret life because most people don't know that plants do interesting things. I will then describe some technological advances in biology and their impact on medicine, agriculture, and conservation biology, and then present an emerging paradigm for a new biology for the 21st century. So Darwin was keenly interested in plants. He was fascinated by the amazing diversity of floral forms in flowering plants. And he performed many pollination experiments, especially in orchids, but also with other plants. And based on these experiments, he concluded that nature abhors perpetual self-fertilization, and that cross-fertilization between the distinct individuals of the same species is highly important for the vigor and fertility of the offspring. Ironically, Darwin was married to his first cousin. That was a custom in the day, and he must have been somewhat concerned about the effect of that on his children, which actually were pretty sickly. So in fact, because plants cannot move to select their mates, they have evolved a number of adaptations that promote outcrossing and reduce the chance of self-fertilization. One type of adaptation is to have female plants and male plants. And an example of that is the date palm, which is very important in this area. And there are plants that have female flowers and plants that have male flowers. And it's been known for a long time, as shown by this image of a Syrian priest pollinating date palm, that if you want to obtain fruit, you have to pollinate the female flowers with pollen from the male plant. Another adaptation for outcrossing is the physical separation of female and male flowers on the same plant. An example of that is maize, where the pollen is produced on the terminal tassels and the eggs or ovules are produced more basally and produce the ear. Of course, maize is the darling of American agriculture, and the reason for that is that it fulfills Darwin's prediction of the hybrid vigor. As you can see from this graph of historical US corn grain yields, up to the 1930s, the yield was stagnant at relatively low levels. And as soon as hybrid corn was introduced, the grain yields shot up. Now, these two adaptations that I just mentioned are only found in a small minority of flowering plants. The majority, about 70% of the 350,000 known species of flowering plants, have so-called perfect flowers, in which the pollen-producing anthers are in close proximity to the female reproductive structure. Yet half of these flowering plants are unable to self-pollinate and self-fertilize. And Darwin described these plants as plants which are completely sterile with their own pollen, but are fertile with that of any other individual of the same species. We now know this phenomenon, as was just mentioned, as self-incompatibility, and this is a natural system for hybridization in plants. So the Nasrallah team decided to study self-incompatibility in this family, the crucifer family, which is so called because all members of this family have flowers in which the petals are arranged in the form of a crucifix. This family includes several economically important plants, such as the vegetable crops of Brassica oleracea, and we all know about cauliflower, cabbage, etc. And all of these were selected by breeders from a wild mustard plant. And this is a fascinating example of the amount of genetic diversity that is harbored by wild plants that breeders can select on. Another important crop is Brassica napus, whose seed is used for the production of edible oil or canola oil and biodiesel. Mustards, the seed that produce condiments, radishes, and a very economically unimportant plant, a weed called Arabidopsis thaliana, which has become the model for plant biology research. So self-incompatibility may be thought of as a self-non-self recognition system. This is a scanning EM picture, which is falsely colorized, that shows the pollen produced in anthers, the female reproductive structure with the terminal stigma consisting of elongated epidermal cells, and the ovary where the ovules or eggs are produced. It is all in self-incompatible members of the crucifer family. It's only when pollinators bring pollen from a genetically unrelated plant of the same species that pollen tubes are allowed to germinate, elongate. Pollen tubes will elongate, reach the ovules, and effect fertilization, and you get seed. On the other hand, if the pollen is derived from the same flower, or another flower on the same plant, or a flower from a genetically related plant, the pollen is inhibited at the surface of these cells. It's unable to produce pollen tubes, and you get no seed. So in this case, self is rejected, and non-self is accepted. And this is the converse of mammalian histocompatibility, which was studied by Peter Mudawar about 70 years ago, in which histocompatibility controls whether a transplant or a graft is going to be accepted or rejected. And in that case, self is accepted and non-self is rejected. So using the techniques of molecular biology and recombinant DNA, we were able to identify two so-called self-recognition genes, one of which encode two proteins, one of which is expressed at the surface of these elongated cells of the stigma, and another one is expressed on the surface of pollen grains. And one can think of those, they're called a receptor and ligand pair of proteins, but one can think of them as a lock and key system with the stigma protein or the receptor being the lock, and the pollen protein, a small peptide of 50 amino acids, which is a key. So this diagram shows how this works. So we have the receptor or the lock displayed at the surface membrane of the stigma epidermal cell, and because these are plants, they have a cell wall. And the key is located on the surface of a pollen grain. So in a self-pollination, the key that's produced by the self-pollen grain fits the lock and will bind to it, activate it, in other words, unlock it, and you have a signaling or a cellular response that leads to changes at the surface here that prevent pollen from hydrating and germinating. On the other hand, in a cross-pollination, the key on a non-self pollen grain does not fit the lock, so it will neither bind nor activate this cellular response, and the pollen tube is formed. So to prove that these two genes actually were required for self-incompatibility and determined self-recognition, we turned to species differences in the genus Arabidopsis. I mentioned Arabidopsis thaliana, and its closest relative, Arabidopsis lyrata, which both these two species diverged from a common ancestor about 5 million years ago. Arabidopsis lyrata is also a wild weed. It is self-incompatible. It's insect-pollinated. It has large flowers and floral scent because it has to attract insects. On the other hand, Arabidopsis thaliana is self-fertile. It self-pollinates without any insect help. It has small flowers and no floral scent because it does not have to attract pollinators. And various lines of evidence have shown that self-incompatibility is the ancestral state in this family. And several times there has been loss of self-incompatibility and production of self-fertility as a result of the loss of one or both of the self-recognition genes that I just mentioned. Self-fertility, this transition is very important because it can lead to reproductive isolation and speciation. So we performed an interspecies gene transfer experiment where we isolated these receptor-ligand gene pair from self-incompatible Arabidopsis lyrata, introduced that into self-fertile Arabidopsis thaliana, and showed that just transformation with these two genes could confer self-incompatibility on these plants that had been separated for 5 million years. So we could restore self-fertile Arabidopsis to its ancestral state of self-incompatibility. And this not only shows that the two genes are actually responsible for self-incompatibility, but it also supports a basic tenet of Darwinian evolution, which is that relatively small changes in genetic makeup can have a profound effect on the diversification of species. So in a field of wild mustard, there may be 100 different stigma receptors, each of which perceives a self-pollen ligand, or the key. And this is analogous to our olfactory system, where there are hundreds of different scent receptors, each of which perceives a different chemical odorant. And this lock and key system is by no means unique to self-incompatibility or to plants. And that's because we now know that plants and animal cells use these type of lock and key systems to perceive signals from the outside of the cell and respond to them. The key can be Depending on the system, the key can be a pathogen-derived factor, a hormone, a growth factor, but in all cases, it will fit its matched lock, and it will activate it and produce a cellular response. Now that we know from genome sequences the full complement of genes found in animals and plants, we know that there are many more such genes that are shared by animals and plants, but they are used in different contexts, biological contexts. So it's clear now that the same genes were repurposed over the course of evolution to function in different processes. Okay, in the next part of my talk, I'm going to describe some recent technological advances that have fueled a revolution in biological sciences. So very quickly, we have DNA synthesis, which now allows a synthesis of very large molecules of DNA, even entire chromosomes, which can then be reintroduced into cells and shown to function. We have DNA sequencing, which allows the reading of entire genomes, and that is all genes in an organism. DNA amplification, which allows, starting from a very small sample, to produce millions of copies of a specific fragment of DNA. Gene transfer, which involves techniques such as microinjection or using the gene gun, which was invented by a Cornell professor. I don't think he was a cowboy. But anyway, this gun actually shoots DNA into cells. And then we have genome editing, the newest technique, which allows very precise changes to be made in the genomes of pretty much any organism. Mass spectroscopy advances now allow defining all proteins encoded in the genome or the proteome and all metabolites made by an organism. Microscopy, advances in microscopy now allow a protein that's tagged with a fluorescent tag to be visualized and tracked in live cells without killing the cells. Advances in cell and tissue culture, which have been in place for plants for over 70 years, where one can start with single cells or pretty much any tissue of a plant and grow it in culture and regenerate whole plants. And also, of course, cell and tissue culture in animal systems, starting with cell cultures, organ cultures, tissue engineering, and now 3D printing, which is shown here by a 3D printed image of an ear from Anthony Atalla's lab at Wake Forest Medical School. Other technological advances have allowed the description of a wiring diagram for the roundworm, C. elegans, which is a model for neuroscience. And this diagram here shows all the connections between the 302 neurons of C. elegans. And this has laid the stage to the so-called Human Connectome project, in which aims to map the connections between all 86 billion neurons in the human brain using MRI, sophisticated MRI methods, microscopy. And this is, of course, part of the most ambitious project in biology, which is to understand everything there is to know about the human brain. But the two most impactful advances have been in computation, which has improved about 25,000-fold in the last 2 decades or so, and DNA sequencing technology, which has improved 25 million-fold within the same 2 decades, and is much cheaper than it used to be. To give you an example of how this change has happened, how dramatic this change is, the first human genome that was sequenced, all 3 billion letters of it, were sequenced, it took 13 years to complete at a cost of $2.7 billion. Now one can sequence a genome within 2 weeks and at a cost of $1,000. So these two developments, computation and DNA sequencing, have launched what is now known as the genomic era. And a quick definition of genomics, it's a branch of molecular genetics that applies recombinant DNA, DNA sequencing, and computational methods to the analysis of genomes. And it also refers to the genome scale technologies and their applications to all areas of biological investigation. So 150 years after Mendel, it's possible to read any genome and describe how species differ from one another and how any two individuals differ from each other. By now, a large number of genomes have been sequenced, and we can expand this sketch of Darwin's tree, which he sketched in a notebook of his in 1837, to encompass the three domains of life: bacteria, archaea, and eukaryota. And this tree here shows our position in the tree on one branch, along with animals, fungi, and plants. And this is an example of species whose genomes have been sequenced in these three branches: fungi, plants, and animals. So, and these are the gene numbers. So you can see that in fungi, the range, depending on the species, is 2,000 to 12,000 genes. In plants, it's 27 to 45,000 genes. In animals, it's 17,000 to 26,000 genes. Now, if we look at human, chimp, and mouse, they all have about 21,000 genes. This came as a big shock, not only because humans have the same number of genes as mice, but because humans have fewer genes than even the smallest plant genome. And this shows that plants are extremely complex. Again, because they can't move, they have to respond to their environment by modifying their biochemistry and physiology. So they have a lot of secondary compound products that are made, many different pathways, and it will take several Professor Haddadines to solve these pathways. If we look closer at these three species here, not only do they have the same number of genes, but they have the same sets of genes. And they're very similar. The genomes are very similar. The genomes of human and chimp differ by, which diverged about 5 to 7 million years ago, differ by about 4%, whereas the genomes of human and mouse, which diverged from a common ancestor about 75 million years ago, differ by 10%. Recently, progress, or not, depends on what recently means. A few years ago, advances in isolation of DNA got so good that it was possible to isolate DNA from fossilized bone samples. And sequencing the Neanderthal genome by now from a first 38,000-year-old fossil and then 50,000-year-old fossil have shown that the Neanderthal genome and the human genome are virtually identical. They separated from a common ancestor about 0.5 million years ago. And in fact, comparison of these genomes shows that humans have 1 to 4% of Neanderthal DNA. So they obviously intermixed. Okay, so now I want to turn to applications of genomics. And there have been many applications of genomics. One that might be of interest to everyone is precision medicine or personalized medicine, in which treatments would be tailored to the genetic makeup of individual patients. Precision medicine holds particular promise for cancer treatment. So in conventional medicine, which is performed now, if patients present with a tumor, let's say, the tumor is analyzed by tissue analysis. The patients, and depending on whether it's a lung cancer, pancreatic cancer, or whatever, the patients are subjected to one treatment, with the result that some patients will show a positive effect, but other patients will show no effect, and some will even show a negative effect. The idea behind precision medicine is that this tumor or cancerous cells will be analyzed not only by tissue analysis, but by DNA, protein, and metabolite profiling. The patients will be separated into groups based on these profiles, and the patients will be given alternative treatments with the hope that all of them will show a positive outcome. Developments in genomics and genome sequencing have allowed the field of metagenomics, where one can sequence whole communities of microorganisms at once. And this has spawned a number of microbiome projects. And this came with the realization that microbes are very important for human health, for the health of plants, and for the health of ecosystems. So the Human Microbiome Project aims to identify all species of microbes that populate our bodies, the mouth, the gut, the skin, the plant and soil microbiomes in health and disease, because that can change. The plant and soil microbiomes aim to identify all species that associate with plants and help them acquire nutrients or allow them to fix nitrogen. And the Earth Microbiome Project aims to identify all microbial species in various ecosystems, oceans, rivers, mountains, below ground, above, up to 10 kilometers above the Earth's surface. Genomics has also practical application in the breeding of crops and livestock. Of course, humans have been genetically modifying crops, plants, and animals ever since the time of domestication by selection of desirable traits about 10,000 to 12,000 years ago. One of the centers of domestication, of course, is the Fertile Crescent, where a number of plant and animal species were first domesticated. And archaeological records show that the transition, at least for wheat and barley, the transition from wild to cultivated took several thousand years to complete. Of course, we now have an accelerated form of crop breeding. In the 20th century application of Mendelian genetics in traditional breeding, where crosses are repeated and individuals selected over many generations, the average time to generate elite cultivars with a desired trait is about 12 years. Now, the application of genomics to so-called precision breeding further accelerates the process, brings it down, at least for now, to about 5 years. Precision breeding using genomics involves whole genome DNA analysis, and it allows accurate prediction of traits from genetic makeup in very young seedlings. You don't need to have whole grown populations of plants, which means that we have a form of remote breeding where a person analyzing the DNA, whole genome DNA data in Japan, let's say, can instruct a breeder in Africa as to what cassava plants to select for further rounds of selection. Now, one problem with domestication and selective breeding is that it creates, it causes loss of genetic diversity because of the formation of population bottlenecks. So a wild progenitor that in domestication has a high degree of genetic diversity, as shown here by these different color circles. As humans started selecting a few individuals with desirable trait, you lose a lot of this genetic diversity, and the domesticate is usually less diverse than the progenitor. Selective breeding using Mendelian genetics aggravated this phenomenon further, and now we have crop varieties that are very uniform genetically. There's further aggravation of this loss of crop diversity due to the practice of monoculture. So in 1903, there were hundreds of varieties of these crops, as shown by the white bars here. By 1983, only few varieties were still cultivated. So about 70% of crop varieties were lost during the last century, which means that cultivated crops have a very narrow genetic base. So we have elite crop cultivars, some of which are grown in Lebanon, that were really developed primarily for the US and European markets. They have low genetic diversity, and because they were developed for these markets, they're not really well adapted to local conditions. So they have a reduced capacity to adapt to fluctuations in the environment, drought, and disease. On the other hand, crop wild relatives and land races that traditional farmers used to cultivate are genetically diverse, they're well adapted to local conditions, and they represent a genetic reservoir of valuable traits. So there's a push now to reintroduce genetic variation for desirable traits from crop wild relatives and land races using whole genome analysis into elite crop cultivars. So we removed them initially, now we have to reintroduce them. A study has shown, has identified regions in the world that are rich in wild relatives of cultivated crops. And this map shows that the Eastern Mediterranean, including Lebanon, is one such region where there are large numbers of these crop wild relatives in one geographic area. And in collaboration, this study was done at the International Center for Tropical Agriculture, or CIAT. And CIAT has partnered with Lebanon to collect the wild relatives of these crops here. And I believe these collections are in progress. But one needs to do more than collect. It's important to utilize this biodiversity and to breed target crops to broaden the local genetic base as an insurance against future environmental stresses. And of course, Lebanese cuisine would be not in good shape if you didn't have chickpeas, lentils, and bread. And these are, of course, very healthy legumes, chickpea and lentils for the healthy Mediterranean diet. But unfortunately, as far as I can tell from just searching, there aren't many, if any, breeding projects underway in Lebanon. Lebanon is also part of the Mediterranean Basin biodiversity hotspot. It's rich in biodiversity relative to its size. Unfortunately, several species are endangered or already extinct. And of course, everybody knows this is due to human-induced land and ecosystem degradation. I won't show any of that at 9 o'clock in the morning. It's very depressing. But this situation is not unique to Lebanon. The entire Mediterranean basin suffers from it. The Mediterranean basin, of course, has been populated for millennia. And it's also a worldwide phenomenon. And some people believe that we are in the throes of a sixth mass extinction. Then there is climate change, drought, and desertification. This image shows the encroachment of the Syrian desert into the northern Bekaa Valley of Lebanon. And a study by NASA showed that 1998 to 2012 was the driest 15 period of the last 900 years. There have been quite a bit of decrease in water storage, including in Lebanon. So all of this creates further threat to biodiversity and crops. And here is a limitation of genomics and breeding. Breeders may be able to use genomics to produce crops that can tolerate drought for maybe a month, but not much longer. So it's critical that measures are put in place for proper management of water resources. There are some efforts in Lebanon at slowing down ecosystem degradation and conserving biodiversity. There are efforts at reforestation, land reclamation, protection, restoration of ecosystems. All of these efforts are useless if we don't have water. And it's also important to expand these efforts, to have a comprehensive description of the status of biological diversity in the country, and then to use conservation genomics, applying genomics method in conservation efforts. Now, one might think that this endeavor would be extremely expensive and impossible to carry out in Lebanon. However, the methods, the way biology is done these days, when it involves sequencing, is the following. Very few labs, even in very affluent countries, have direct access to high-throughput genome sequencing facilities. So what one does is you prepare your DNA sample, you ship it to a central DNA sequencing facility. They produce the sequences, ship back the data to you, and then what is done by the user is to analyze and interpret the data. So what one needs is not an expensive sequencing facility, but the training of individuals who have the capacity to analyze and interpret the data. the large sets of data that are generated by genomics. So what of biological sciences in the 21st century? Genomics will certainly remain central to biological sciences in terms of helping to solve many of the biological phenomena that remain unsolved. Health and agriculture, as I mentioned, ecology and conservation. So the training of biologists for the 21st century should involve or should provide a deep understanding of new genetic and genomic technologies and their applications. It should also provide, as I just mentioned, essential skills in computational biology or bioinformatics to analyze and interpret very large data sets. It's important also in education and outreach to provide the citizens with environmental sustainability education and increased science literacy in the public at large. And this is especially important because of the profound ethical, legal, and social implications of new genetic and genomic technologies. In particular, I'd like to single out genome editing, which raises profound and very disturbing questions regarding the possibility of scientists permanently introducing permanent changes in the human genome that are heritable. So one has to be careful. It's not all rosy. Okay, so to end, I want to present an emerging new biology paradigm for the 21st century. The new biology will still be grounded in biology, but it will integrate information from many different fields, okay, from social sciences, physical, chemical sciences, computer science, math, science education, engineering. The goal is that this scientific integration will produce a deeper understanding of biological systems, which in turn would lead to biology-based solutions to societal problems in health, environment, energy, and food. So who is the new biologist? The new biologist is not someone who knows a little bit about many different fields. It's someone who has a deep knowledge of one subject and a working knowledge in one or more other topics. For example, an engineer who designs systems for sustainable use of resources that are essential for ecosystems is a new biologist. A mathematician who derives equations to explain the complex interactions that characterize cells and all biological organisms and their ecosystems is a new biologist. A physicist who tries to determine how the laws of physics apply to the complex and non-equilibrium condition of living cells is also a new scientist, a new biologist. So training the new biologist will require integration, a multidisciplinary training approach where many of the students are exposed to many of these. And I think that universities such as AUB, and particularly the Faculty of Arts and Sciences, is well positioned to provide such interdisciplinary training. I would like to close with this painting of Paul Gauguin. In this painting, which is the largest one he painted, in the corner he wrote 3 questions. D'où venons-nous? Que sommes-nous? Où allons-nous? Genomics and biology has started to provide some answers to the first 2 questions. The answer to the third question depends on what we do next. And I thank you for your attention.